Good morning, everyone. I welcome you all to this 8th day of CSIR online training. Uh, am, am I audible? Please respond in chat box because then only we can start the session. Am I audible? Can you see my slides? Yeah, okay. Now start the section. Okay. Today is the 8th day. We are going to discuss about the morphogenesis and organogenesis. Previous day we discussed about developmental biology, how the cell develop into the tissues. Tissues are how it is differentiated based on the cell signaling. Today's section is going to discuss about the morphogenesis and organogenesis. Okay, morphogenesis is fully responsible for the structure formation of the particular organ or particular uh, uh, living thing. Like that may be the bird, that may be the mammalian cell, that may be the bird. Okay. Then organogenesis, each and every organs are developed after the determination process. So the how the cells are yesterday we discussed the gastrulation. After that gastrulation process, how they developed into organ. These are the studies are we are going to discuss in the today class. And these are all the topics. First we are going to discussing the morphogenesis of and organogenesis in the animals. In the animals, different animals having different process in morphogenesis in organogenesis. Here we are going to discuss about the Dictyostelium and Amphibia, then chick. Okay. These are the some examples for morphogenesis and organogenesis in the animal. Then we in the plant we are going to discuss about the how the root and shoot development happen in cell development. Okay, what is morphogenesis? Morphogenesis is a formation of ordered form. Okay, if in a human baby formation, each and every parts are formed based on the programmed way. Okay, first this organ want to develop, then this organ want to develop. That kind of process, step by step ordered process. The formation order is not changeable at any time. In the morphogenesis, the order of formation is very important. In the animal, you have a 4 different morphogenesis process: cell division, cell shape and size, cell death, and cell migration. Okay, in the 4 steps are the for tissues and cells are forming organs for this the organs based on this 4 step. In a plant, that migration is not there in the process. Only cell division, shape and size, and cell death happen. These are all the morphogenic steps process involved in the cell development. Then in the here it's an one example. They are explaining with a step. Okay, during the morphogenesis, the gastrulation and organogenesis are the 2 different steps involved in the particular morphogenesis process in animals. Okay, gastrulation we discussed in the earlier class. Then organogenesis process of initiation and development of structure. Okay, this in this organogenesis and morphogenesis, cells want to be developed in an ordered manner. Okay, this eye cells want to develop in this sites, kidney cells want to develop in this size. Apart from this, that cells don't allow to grow over the particular fixed size. Okay, that is very important thing happen in the organogenesis and morphogenesis. In the CSIR examination, many of the questions are based on the cell count and cell size. They are providing the they provided the question with the eye cell development, that lens development. That kind of questions we want to understand the concept and try to find out the answer based on their basic concept involved in the organogenesis and morphogenesis. Okay, it's the ability of metristomatic plant tissues from the various organs in de novo. It's de novo is production of cells in the de novo way. That production may be forming the shoot and root. We can develop the shoot and root in laboratory scale itself. Just collect the samples, collect the cells from the plant and grow it in a specific medium in laboratory. Then we can develop the shoot and root. We can develop the leaves also. These are the each and in the plant, that is very easy. In animal also, it's experimentally proved. We can prove as a it can be possible. Okay, they are in the laboratory scale. They develop the animal ear, that mammalian cell ear, heart tissues, hearts formation. It's a current trending research ongoing in the various research laboratories. Here they mentioned plant generation, and it's similar to the human also, the mammalian system also, carrying the organogenesis, the psychotic embryo culturing, somatic embryogenesis, and the organogenesis. These are all the steps involved in the that mammalian cell culturing systems. Another one thing, embryo can't able to develop as a organ. Embryo is want to develop as a blastula then forming the various organs. That very important part. Embryo can't able to develop into organs. That need one that need a basic determination step. That determination step forms the blastula. That blastula it's responsible for production of various organs. Okay, it's very it's a basic image, but on this image they are having n number of question based on this image. So only I am including this image, because that each and every cells are responsible for specific organs, right? Okay, here first one is the activin A. It's an medium for cell cultivation. That in that cultivation medium we can cultivate the mesenterodrome. This mesenterodrome is having the signaling molecule on the surface. If they receiving the Wnt3a signal and FGF4 signaling, they are forming the hindgut endoterm. Otherwise they are receiving the BMP4 or FGF9 signaling, means that are forming the intermediate mesoterm cells. Okay, here it's very important concept, the mesoterm and mesoterm formation. This mesoterm intermediate, after that this intermediate mesoderm, that cells are developed into kidney cells, kidney organoids. Okay. Okay, here question, how they ask a question means, okay, order of the cell production, after order of the cell production in kidney cells. They are given various given the steps in various order. We want to understand the basic concepts. So mesoderms are receiving this, if they are receiving a BMP4 and FGF9 signaling, that forming the kidney cells. Otherwise, if they are receiving the Vent3A and FGF4 signal, means that form as the intestinal organoids. Both are present in the in the stomach, that formed based on the signal variation. It's a one thing, very important thing happen in a cell tissue development. Okay, then in the laboratory scale, it's a laboratory scale experiment. Likewise, that it happen in a human body. Another thing is a minimal medium. If they are using the minimal medium is specifically designed for the human cell culturing. That is, if that may be the human cell culture, that may be the mammalian cell culturing. Here, ectoderm. Ectoderm is, yesterday we discussed, right? Mesoderm, ectoderm, endoderm. And if in the exoderm is only the outer cell, outer tissues, skin formation. So mesoderm and endoderms are helpful for the production of internal organs. The mesoderm is specifically for internally intestinal and kidney, liver, that kind of heart, that production. Ectoderm is the outer layer of the outer layer, means internally present over the mesoderm. If they are receiving the 20% of KSR is a medium, they are that are producing the neuroepithelium cells that helps to cerebral organoids. Okay, ectoderm helps to cerebral, if they are producing the neuroepithelium, that have a ability to produce cerebral organoids. If they are producing the retinal epithelium, that producing the optic cup organoids. Okay, can you understand here? Here it's an eyeball. I think it's understandable. Here it's most repeated questions, like this kind of questions: cell culture medium, cell culture tissues, and organ development. Here we have on extra the one big table. In that table they mention many cells and the organs. Okay, I'll send the reference with your mail IDs or Google Class. It's a one small explanation about the organogenesis. Here first one is the neural curve, neural curve formed on the cell, that is the gastrulation formation. After that, cells are forming the own cavity inside the nerve tube. It's a exoderm. Here that's a mesoderm is formed. Okay, first previously we are understand what is the morphogenesis, what is the organogenesis, how it has happened. Now we are entered into the CSIR exam topic. In the exam topic, they mention organogenesis and morphogenesis of Dictyostelium. In the Dictyostelium, it's a fungi. It's a very, very small fungi. It's look like it's only seenable in the mic. Where many of the researchers are focused on the same image. It's an fungi. It's spend most of time in the vegetative state. Vegetative state means that is doing the metastatic mitosis division and divided into various tissues that formed in, they are duplicating, formation of duplicating. and if they are receiving the food from the bacteria and the minimum soil foliage leaves, that able to produce the sexual cycle. It's having two different cycles. One is vegetative and another is a non-vegetative cell. Here the vegetative pathway is a regular pathway of that fungi. Vegetative means that can be involved in the mitosis. During the mitosis, the cells can be divided into two. That divided after the division, that are again undergone the mitosis. The process repeatedly happen in the cell cycle. It's developing in the generation. Sometimes it's if they're receiving the foods from the bacteria that are able to produce the involved in the sexual cycle. That is here they mention. It's a very this fungi comes under the metazoa branch. Just the explanation, first one. Okay, next is the pictorial image of our this compound. Okay, wait 1 minute. Okay, it's the best example. Here first one is a vegetative cell. Here 0th hour, we can see here 0th hour we have the vegetative cell. That vegetative cell is a normal vegetative cell cycle produced cells. These cells are after 4 or 5th hour, it's forming the that are communicate between each other. Okay, we discussed in earlier classes, bacteria is having the quorum sensing. Quorum sensing means that having the cell communication based on the cell surface receptors. That quorum sensing is a very important concept in the bacterial cell cultivation. This bacterial cell that bacteria are communicating with the fungi too. That is forming the quorum sensing in the fungi also, that communicate with each one between 1 to 1 cell communication. So that are forming the aggregation. In a regular life you may seen in the fungi, right? Fungi are forming the network. That network is called as the aggregation. It happens in the 8 to 10th hour. Okay, after getting the aggregation, that forming the mold. Just imagine what happened in the bread. First there was the some small quantity of fungi are formed. Then after getting more signals from in the signals between them, that forming the mold that become big in size. That mold is produced in the 12th hour. After but here I'm just to tell the fungi for example. Here after you want to change the concept. After that mold formation, that forming the pre-stalk cells. These pre-stalk cells are forming or on extension mold structure. The mold structure is in a molded form that are having the pre-stalk cells. These pre-stalk cells are producing some external development on the particular mold. It happened after the 14th hour. It's then that having two different cells in the cell structure, so it's forming develop the slug. Okay, slug that happen in the 16th hour. This red color zone is the specifically explained as morphogenic process. That is formation of slug and Mexican hat formation, culminant formation. This is happen in the morphogenesis. Here they ask the question, how the vegetative cell forming they ask the step-by-step process. First one is the after vegetative process, aggregation, mound formation, slug formation, then Mexican hat and culminant. and the another chance of question and that fruiting body formation, that after the fruiting body formation the egg having the spores that releasing the spores in the environment, that spores can be able to produce the microbes. These are the step by step process. Here itself they mention the same concept. Okay, first is a individual cell, this is a growth the first the small size round is the vegetative pathway. After this vegetative pathway it's forming the aggregation, it's forming the slug. In the slug that having the prostock and pre-stock cells, that accumulation, that is the hat, the Mexican hat formation, then fruiting body formation. Okay, it's the same process. For understanding I am including the same concept for multiple image. Okay, I'm including the multiple images for the same concept, because for understanding only I am included this. Okay, this is the basic one. First it's 10th are the mold formation. After the mold formation it's forming the small external development, then slug formation, hat formation. After hat formation it's develop as a culminant. It's then developing the fruiting body. This fruiting body is after particular sometimes it's after 24 hours it's removing the spore. That spore is developed spread into air. It's a basic understanding image. It's really naturally captured by the SEM. Here we discuss the story about what happened in the Dictyostelium cells. Now we are going to discuss about what is the cell signalings happen in the Dictyostelium cell development, and then the morphogenesis, organogenesis process. It's a basic one. Here it's fully depends on the G coupled protein receptor cell signaling. That signaling makes some the activating some pathway, that activation of the F actin pathway, that is the anterior production. If they are activating the myelocells, that activating the myelo, that suppression of latent pseudopodia formation is suppressed. Okay, it's then another. First we discussed the Dictyostelium development. Now we are going to discuss the Drosophila development. This concept we studied in 12th standard itself, but we may forget. Just I am refreshing the concept. How the cell adult, how the Drosophila developed from the cells to that segmented formation, everything. Okay, first one is a fertilized egg, that zygote having the nucleus of nucleus of the Drosophila. The anterior and formation, that is, this is called as the first edge, the enter of this after the fertilization, the egg is developing four. One is the anterior and the posterior, ventral and dorsal. The four parts are the majorly role playing parts, the directions, because based on this concept only we are going to the anterior, posterior and the dorsal, ventral. Okay, here 9 nuclear division, the syncytial blastoderm. Syncytial blastoderm means the present in the in this stage are forming the syncytial blastoderm. That syncytial blastoderm means the cell communication happen in between the small area. So the cells, here you can see the cells are arranged, cells are developed. After the fifth hour, the cellular blastoderm is formed. That strong cells are formed in the cell. Here, after the cellular blastoderm formation, the protein gradient, we yesterday discussed in on slide, I think, the protein gradient establishment segmentation. Based on the gradient, that cells are developed as a different portion, different organs. So based on the gradient of protein, that developing the various segmentation in them. Here you have the two different region, thoracic segmentation and abdominal segmentation. The thoracic is a front portion of the Drosophila. Abdominal is a back portion that having the signal different metamorphosis. That metamorphosis happen in earlier stage of the embryo development. Upcoming slide I will discuss about the metamorphosis. That here it's a pattern formation. That is the okay, this place we want to segmentation, this place we want to thoracis, and this place we want to head. It's the pattern formation. This pattern formation is the cytoskeleton imposed often symmetric on the egg. Based on the egg formation, that forming the cytoskeleton. The gradient of the soluble proteins are laid down among the A to P. A to P and D to V. A to P means the anterior and posterior. D to V means here they are dissect like here it's here dissect like this, anterior to posterior and dorsal to ventral. Based on this gradient, we developing the various organs leading to the broad region of gene expression, that based on the brief gene expression alone. Okay, the axis development. Axis development is a important thing in the Drosophila, because that having the different structural parts that want to express in a specific way. Here in the structure of the Drosophila, we have the different segments, that is the patterns. A is antennapedia complex. This antennapedia complex are having the 8 segments, and the pithorax complex, that pithorax complex also having the different portions. Based on the portions, they developing the expression. Here the LAB gene is responsible for the whole cell segment for pattern forming. This after the pattern forming, the 1 and 2, every here they have the portions, right? T1, T2, T3, A1, A2, A3 to A8. This and other likewise here 8A is here. It's an it is the one side of the Drosophila. Another side of the Drosophila also having the same pattern, that is called as the that is also having similar numbering. So that is the 8 to 14, 9 to 14. Then the DFT. These words are very very important, because they are asking the question. Okay, LAB. I most repeatedly I have seen the word the LAB, because the concept of pattern formation is very important topic in the Drosophila cell development. So only I am mentioning this. LAB is responsible for the pattern formation, that is the labial. Then DFT, deformed means okay, that is getting after the small pattern forming, that want to differentiate, that is a deformed. Then SRC, the sex combo reduce, it's developing the particular portion that pattern into the regions sex combo reduce. Then Antennapedia is a very important thing happen in cell development of Drosophila. Here that developing particular organs. Then Ultrapithorox, this Ultrapithorox is a development of some organs in Drosophila. Then another is abdominal A, abdominal B. That organs are separately developed. These are the whole step. They asking the question based on the gene and the portion. Okay, they providing the gene name and asking the organ development. These are the questions are most rapidly asked in the CSIR examination. Okay, maternal gene. Here this is the previous slide I mentioned, right? The dorsal, anterior, posterior, ventral. Here the mention here the anterior, that anterior development of head and thorax. Main thing, the anterior development is the development of head and thorax. Posterior is the development of abdominal segmentations. Terminal system, that is the terminal system is development of specialized structure of the particular species. Dorsal ventral is development of the transmembrane receptors. Please understand the words. The maternal genes having four different axial words, that is the anterior system. The anterior system helps to develop the head and the thorax. Then the posterior system helps to develop the abdominal gradient. The terminal system helps to the specialized structure. The dorsal and ventral system helps to develop the transmembrane receptors. Okay, how they happen? Posterior and anterior axis. If the bicoid that having the anterior, because the low concentration of having the low concentration of particular anterior and the higher concentration of the mRNA molecule, and it's it having the two concepts. Here the small that in this image may explain you very well how the embryo will positionate in the cell system. If that means the bicoids means that previously that at a low concentration itself they are in the locoid, that low concentration they can't able to survive, so that embryo is need a higher concentration of the proteins. Then in the likewise they have mentioned the hatchback, the caudal and nanos. These are the four concepts in the anterior and posterior axis. This anterior and posterior axis are helps developing the intestinal organs and the head formation. It's again example. If here they mention, right, weaker gradient, the gradient means the protein gradient formation. If there is a weaker gradient, the anterior part of the embryo looks more posterior. it's not there is no different. Okay, here for example, in a snake we can't find the difference between the we can't find that much difference between the head and tail, so that is the weaker gradient. There is no organal development in the in the head. Then another example is the worms. Worms don't have the difference between head and intestine, in head and some other parts. So that is the weaker gradient. Stronger gradient are human body. Have a strong difference between the head and intestinal part. Some of the unfunctional picoids are there that are the converted into the abdomen, that making the embryo with the posterior structure. Here this is the gradient, how the gradient works with the embryo. In the earlier stage there was the gap. Here they have the 4 different stages. Oh no, sorry, wait, 6 different stages. In the 6 different stages the various process happen. First one is the gap gene. Gap gene is a process which held in the earlier stage of embryogenesis. In this step there are several adjustment segmentation missing. Okay, here it's an uneven segmentation, right? In a normal cell that we want a fixed gradient, but it's abnormal. That define 4 broad regions are in the present in the egg. Another one is the pair rule gene. Pair rule gene means the even numbered and odd number segmentations are missing. Okay, only one thing here, the if the light color means that is odd, dark color means that's even. For example I am taken the 1/2 of the number of segmentation. Some portions are missing in the pair rule gene. Okay, only on either there is the higher expression of the even numbered segmentation, or higher expression of the odd number segmentation. Then next is the segment polarity gene. Segment polarity gene means, okay, we after getting the information from the pair rule gene, they are understand the okay, this much of stripes we want to make in the cell surface. So that step is the segment polarity gene. After segmentation formation, that is after fifth or the cellular blastoderm, that is the forming the cellular blastoderm. This cellular blastoderm is having different partitioning. That partitioning is that development, so partition development. The groove get deeper and more. Here the in the you can see in many ups and downs on the Drosophila cell surface that has formed between the 9th and 10th hour. After the development of the adult fly, that after the development of embryo and segment with the development, that is called as the adult fly. That adult fly has the 3 thoracic segment and 8 abdominal segment. It's a very important term. Adult fly having the 3 thoracic segment and 8 abdominal segment. They confused in the question. Okay, they having the 14 because we earlier we studied 14s, right? That is the count. That is the count of the both side. But the question, the answer for the question is the 8 abdominal segmentation alone. Very important thing, you want to keep it in mind. Adult fly has the only 3 thoracic segmentation and 8 abdominal segmentation. Again I am explaining with the image alone. Here this is a pattern formation, the same process. For understanding I am including, because yesterday some of them mentioned we can't understand the process with one image, so I am including the different images for understanding. Here okay, in the gene it's for example Here that there is a no gradient formed in the particular cell, embryonic cell. After this gradient formation, based on the gradient, that developing the different segmentation in the cell. It's a cut down image of anterior and posterior. Okay, here it's a hemotic gene, so that control the final development. This is the color, the head want to be in color, in this color, that the body want to be this color, and the abdominal want to be in this color. It's a fixed one, the hematopoietic cell, sorry, hemotic cell. Then segmentation gene, this segmentation genes are helps to, okay, we the head want that it having only two colors, the control of development of segmentation. Another is a maternal effect gene. Maternal effect gene, it casts the signal, it casts the changes in the maternal, the gene expression, the gene development, DNA expression level. Okay. Likewise, previously we discussed the story about the Drosophila development. Now this is the proteins involved in the Drosophila development. In the Drosophila development, they having the toll-like, if they first they developing the toll-like receptor on the cell surface, that is the endoterm that producing the toll-like receptors, that forming that receptors forming the tube-like structures. This tube-like structure is formed as a, that is the forms the then that is developed into DIF, or it's called a catechus or trocel. If they are in the mammalian cell, is the same happen in the mammalian cell. Interleukin 1, that interleukin 1 is transforming the signal to the that INF alpha, INF kappa B, it's NF kappa B pathway inflammation. This is the ventral patterning. Ventral patterning is one another important concept in the Drosophila cell development. In the ventral concept, we have the different step. If there is a twist on the, that twist forming three different proteins, that is the T-MET2, Thymin and backpi. These three are forming the different responsible for the different process. DMET2 is helps to cell muscle differentiation. Thymin helps to develop the heart development, develop the heart. Backpipe helps to the the visceral muscle development. These are the happen because of the twist on the Drosophila. If there is the in snail shaped means that forming the neuro and ectoderm. Ectoderm means internal one, neuro ectoderm gene. If there is in a rheumatoid shape, that not able to producing any organs. And another is a tolloid, in that's a tolloid, that can be for incubating, that is the incubated structure incubated, structure incubated. We can't develop with the process. So these are all the major ventral patterning. If there is a twist, we can develop the organs, otherwise we can't. If the snail shape helps to produce the neuronal ectoderm gene. This the two concept. If there is an tolloid or a rhomboid shape, and in that particular stage, we can't develop into the Drosophila development. Okay, next we are enter into the Amphibian. Amphibian is a frog development. First we discuss the Dictyostelium, that is fungi. Next we discussed the Drosophila, it's a fly. Next in this slide we are going to discuss about the Amphibian. Amphibian are the, in the Amphibian, the axis formation is a very important thing. Here we have a two different step. One is the first signal, it's tell they are drossel. The second signal is the they tell them okay, it's a mesoterm. This is the two different stages. Okay, here I think this image is visible to you. We can discuss with the image with a clear idea. The metamorphosis is a technical term to explain the cell development. First step is the egg development. After the egg development, after the egg production, that forming that eggs are interlinked and forming the embryo. The embryo after the embryo formation, the tadpole cling. Okay, the tadpole clinging to water plant. They are attached on the surface of the water plant. That produced embryo attached on the plant. That then after the particular period, that forming one tail and head structure. That external gills for breathing. They need oxygen in the water, so their external gills production for the breathing. Then tail keeps on going. So for their surveillance, the tail is continuously grown. Then hind legs appears. After the particular period, the tail, from the tail they are formed the hind legs. Hind legs means the back legs. This legs are they are having lives from food stored in the tail. Because that is the tail which having the intestine also, so that is stored in the tail. The front leg appearance. After the particular period, the front leg will appear. Okay, here we want to understand. First tail is formed, then back leg form, then intestinal storage form, and then only the front leg form. The tail become shorter after the maturation. That tail automatically shorter. Here next one is the younger frog, then adult frog. These are all these tech. Okay, we are all know about this cell development in early stage. Why you are repeatedly telling in the this concept means because Drosophila and this amphibian concept is most repeatedly asked concept in a CSIR examination. Here they are asking they are making some examples. They are making the experiments with this amphibians. So the step by step process is a very important thing we want to be remember. It's another pictorial representation. I am mentioning it for understanding. Here another important thing is hormones involved in the metamorphosis of the amphibians. Okay, thyroxine, triiodothyroxine. These two compounds are mostly involved hormone T4 and T3. Thyroxine, triiodothyroxine. This hormones helps to growth, death, and remodeling, respecification in each and every step they are used. In using this hormone, there are some pathways are activated in the amphibians. That pathways are helps to regressive, progressive, and modification. Regressive means that forming the teeth, gills, tail, that is that formation. Then progressive, in the progressive means the development. In the progressive phase, the limbs, the bunny skull, the tongue, that are formed in the amphibians. It's one major explained image of the cell development in the amphibian. Okay, here it's a normal embryo cell. It having the internal gradient with different angulars, ventral, dorsal, the vegetal and animal. Now these portions are want to be developed to various part. First, the under part is the endoderm, that external surface having the ectoderm, that is the skin formation. Now the mesoderm, that is responsible for the backbone, that internal strong gap that are helps okay understand the this want to be forebrain, this want to be the somatic mesoderm, this want to be the neural tube, that are all the developed based on the mesoderm activity. Over the mesoderm, these are all not modified, we can't modifying this arrangement, because it's a naturally happen, it's a naturally fixed one, we can't rearrange this mesoderm endoderm portion. Here that I mentioned, right, that for two hormones are involved in the process, that's the thyroxine and T4 and T3. These are all helpful for the processing of this flow chart. Here they are based on the temperature. The main thing is temperature, light and nutrition. Without light, the lava can't not develop into frog. Okay, that's a very important thing, that hypothalamus, pituitary and thyroid hormones are segregated in the embryonic cell itself, then based on the T3 T4 that develop into various parts. Okay, after the embryonic stage, prolactin. Okay, prolactin, very important thing. After the embryonic cell development, the prolactin is developed into the responsible for the development of the tail formation. After the embryo stage, the prolactin helps for the tail development. After the tail development, T3 and T4 helps for the organ development and head development. Okay, this another example for the ventral and mesoderm formation. The beta catenin is nodal related high compound. If that is high in embryo, that helps for the organal organization. If that VGT and VG1 proteins there, means that helps to the gastrulation of beta catenin signaling, that helps to the ventral mesoderm. That's like some explanation which helps to understand how the mesoderm endoderms are developed. If there is a VHT and VHT1, means that helps for the gastrulation of beta catenin. Here now we are going to discuss about the chick formation, chick development. We are all previously know about this. I think here first step is here itself we have the anterior, posterior, distal and proximal. Extra embryonic space and embryonic space. This is the two compounds, the extra embryonic and embryonic. In the embryonic stage, having the mesoderm, endoderm, ectoderm all happen in the inside, that helps to develop the organs in the chick. Then it's a bird, the chick also bird, it's also the same concept. Here the duck embryos, it's a duck embryo, its transplantation, the Tuckens mode, that is embryo formation. Then gastrulation formation. After the gastro formation, that forming the backbone. Here the lava formation. Lava formation is very important in the birds and in the cell development, animal cell development. The secondary lava cells, primary lava cells, and secondary lava cell fates. Okay. Here we having the different counts: a 7 cell, 8 cell, and 7 cell. That helps for the lava. Here they are mentioning the it's an very important concept in the volvos development. That volvo development, we have the larger larva stage. That the larger larva stage we having early larva stage we having the cell development from the basement membrane. This that's basement membrane are having a different proteins. The proteins are responsible for the accumulation of the lateral lava stage. Okay, this in this stage we having the ligand receptors concept. Once the signal pass to the epidermal growth factor receptor, they are receiving the LET23 ligand. The epidermal growth factor receptor is if receiving the LET ligand, that faster signals to RAS pathway. The RAS pathway activation helps to activating the MAP kinase pathway. If the MAP kinase pathway is activated, the ETS transcription factor is activated in the cell. This helps to ETS transcription factor helps to the ulval gene that helps to produce the external the internal organs. The next concept is the eye formation. In the eye formation, we have the 4 different steps. First one is the normal induction of lens by optical vesicles. Another one is the optical vesicles can't induce the ectoderm. Okay, we can't induce the ectoderm. The optic vessels is the removed. Now the lens is induced. Then tissues, other optic vesicles is implanted. No induction of occur. No induction is there. Okay, so basic example. First there is an normal induction. Another one is truncated one. Another one is optical vesicles is removed. Then induce this concept. First is this is the step-by-step process. First, the surface. One minute. Please wait 1 minute. Okay. In this eye lens induction concept, the surface endoderm, internal yellow color is the mesoderm. Another one is the neuroepithelium, that is the internal part. Here we want to develop an organ, the lens. So there was some gap is formed in the lens placoid that is induced by the previous step, previously mentioned, right? That is induced by the there was a gap is formed. Okay, there here the gap is formed, but how that lens can be developed? The lens vesicles, it's a gap is formed. Then neuroretinal, it helps to form this lens. The ganglion cells are the major role playing compound in the lens development. The intraneurons and photoreceptor cells, these are all they involved in the cell lens development. It's another highly explained image. In this image they are explained very clearly. First, neuronal ectoderm. This neuronal ectoderm is helped forming the the epidermis. This epidermis is having the epidermis, the internal portion of the epidermis. We have the neuronal cells, right? That neuronal cells ectoderm having one gap and the particular portion. This gap is closed with the preauricular mesenchyme. Preauricular mesenchyme is a very important term in the organelle development, lens development. Then another one is the surface ectoderm. This surface ectoderm is the helps to place the lens placard. Then lens is placed on the placed over the optical cup. Then the chimera cells are covering over the lens. These are the step-by-step process. Then lymph bud development. In the lymph bud development, we have the different stage pattern forming. That lymph bud formed after the 4th week of the embryo. This numerous genes are involved in the lymph bud. In this lymph bud, the DPXA, DPX5 and HOX cluster is two important genes are involved in the lymph development. The T box containing TB is a T box containing the transcription factor, and HOX, this cluster helpful to the lymph formation. The transcription process factors helps to develop the protein. The lymph formation helps to develop the clusters. Another one, so this is the lymph formation. In this I am going to explain with the different segregating the position where the lymph field is generated, and upper bud development and lower bud development. And lymph cells are visible to by the 25th in the you may seen many may seen in any that scanning report of the pregnant women. They are they can seen image in the 26th and 27th day. Here in this lymph formation, and it's a lymph development, that is the hand development, hand or foot development. Here this happen over the human. Okay, first after the development of this shape in the upper layer of it's upper limb bud, right? Upper limb bud, lower limb bud. It is like this. We have it in both sides. This upper limb bud and lower limb bud producing the when the upper limb bud is removed, the limb developed the crisis. When the extra AER is grafted near the existing limb, that is developing the fingers. Once the limb is developing, that forming the upper limb is forming the hand. In that lymph, that may be having the extra AERs, that means that is then the anterior exoderms, means that helps to producing the wings. That may be the wing, that may be the fingers. In the leg also we having the same lymph development process. The lymph means nothing but the hands and leg development, wings development, that's all. Then neuronal development. After the hand and eye development, leg development and neuronal system development, now we are going to discuss. Don't mistake me, because this is a vast topic, but I want to clear about the concept, so only I am taking much time. Just give a 5 to 10 minutes, please wait. Within 5 to 10 minutes I will finish it in a fast manner. In the neuronal development, we have the ectoderm differentiation. In this ectoderm differentiation, we have the epidermis, nerve tissues and neuronal crystal cells. Here this is a neuronal cell development. In this neuronal cell development, it's an axon. First one is the oligodendrocytes. This oligodendrocytes are producing axons. These axons are having the node of the Raynaud's. This node of Raynaud's having the cell. In the node of Raynaud's, they are having the cell, the scatchworm cells. These are all the passing the signals. Axons covered with the other node of Raynaud's. That mode of Raynaud's having the scatchworm cells. These are this production. This is the microscopic image. The post embryo. After the embryo development, we have the three different portions. That is the tissue generation, aging. These are all the post metamorphosis. That is the end stage, end process. This is already discussed. It's all about the maturation. Okay, egg, larva, and pupa is the first embryo to development stage. That butterfly is a post embryogenesis. Okay, here in the sexual asexual production, we have two different parts: the primary and secondary sexual differentiation, unisexual differentiation, bisexual differentiation. Here it's another important topic in the sex determination. In sex determination in human, we are discussing the generally XY XX. If there is a female, the XX chromosomes have present in there. If there is a male, that they have the XY chromosomes. Here in the female they don't have the Y chromosome. This is difference between the 7th week. We can see the whether the child is the boy or girl. It's after the 7th week. In sex determination in Drosophila, we have the same XY concept, but how we can determine means they are producing the chromosomal count change. We are using that only we can find out that concept. That environmental determination, we having the 4 different characters that vary between the temperature difference between the egg development and the pathways, genetic control. These are all the major things happen in the environmental determination. Okay, it's a very important table. Please listen. In the Drosophila, the transduction cascades are regulating the mRNA slicing. That mRNA slicing is targeting the DSX, that helps to key male target gene. If you calculated this means, easily we can find whether it is a female or male. In the elegant we can the A ratio is segregated ligand membrane receptor pathway, MAB3. If there is a MAB pathway protein is there, that is we can be determined that is a male. In a mammalian, we have the SRY dominant. Here in Drosophila and elegant, they having the X and A in equal in some ratio. So based on the ratio, they are producing the protein. But in the mammalian cells, the dominance of Y, we can determine the sex. That because of the hormone, the DMRT1. DMRT1 is a major protein which responds for the hormonal production. That hormone responds for the dominancy of Y. These are the sex determination happen in the Drosophila. Then the environmental regulation in animals are explained with the blood egg development, metamorphosis, this concept. It's a basic thing. Okay, next enter into the plant development. Just one. Sorry for taking too much of time, but we want to cover the whole part in 30 days. It's very difficult, so only I am a rush of this. It's a shoot development. In the plant, we have two different. One is that organ cell development. In animal, we discussed the eye development, neuronal development, limb development, post generation, and formation, pattern formation, axis formation, everything. But in a plant, we have only shoot formation and root formation, leaf formation. That after the shoot and root, that leaf is the common fact. Okay. In here they mention the word what is means apical meristem, apical meristem. Okay. The repeatedly they asking the word in the CSIR question. Okay. Related to this apical meristem, why they are asking? It's a very important for the shoot and root development. It's a midpoint. Okay, it's different undifferentiated cell at a tip of the plant root. Okay, this helps and tip of the cells that divide and enable the plant to grow length for the growth. This apical meristem, we have the primary meristem, ground meristem, and procambium. If there is a primary meristem that producing primary tissues, and if there is a meristem, that ground tissues are the cortex, the carcambium, and cork and pilotum. These are the production of the cork and pilum from the apical meristem. Another is the vascular cambium. The vascular cambium helpful for secondary phylum and secondary xylem. Secondary phloem and secondary xylems are produced from procambium. The procambium is another one classification of the apical meristem. It's an example. The prototerm is prototerm and ground meristem and procambium. This prototerm helps for the epidermis. Ground, it's a very important term in the cell development of plant. Okay. In this portion, we have three different stages. The proterm is help for epidermis, the ground meristem help for the cortex, the procambium helps for the vascular cylinders. In the vascular cylinders is the stem, right? The stem is the vascular cylinder. We have the pre-cycles, that is a carcambium. That carcambium helps for the cork and pilum. The phlotem. Then another one is the vascular cambium. Carcambium is the center part, and the another part is vascular external vascular cambium. This external vascular cambium having the secondary and phloem, primary phloem. Okay, it's a shoot and root development. Its midpoint is called as the apical meristem. This is the pathway which explaining the root and shoot development. In this root and shoot development, the major thing is in the root development, the concentration of axin. It's a the concentration of the proteins alone vary in the root and shoot development. In the root development, we having the root hair development, root branching. In the shoot development, we having the leaf expansion and shoot branching. Stages of leaf development, we have three different organogenesis development of subogen domains, that then is cell and tissues differentiation. In the organogenesis, it's a leaf development, and development of subogen domains is the lateral, proximal, that kind of things. Another one in cell tissues differentiation, L1 layer forms epidermal, L2 layer forms the photosynthesis mesocells, L3 layers forms the vascular elements. These are the leaf development. Here we have the portions. It's the basic cell development stage. P2 is the primordium. P3 is the primordium begins to flatten. P1 is the most recently formed primordium, symmetrical at this stage, because every plant want to be in straight, right? So for that we want to maintaining the axial arrangement. Here are the L1, L2, L3. L1 is the epidermis, L2 is the ground tissues, L3 is the vasculatures. That after the development of the plant, we want to understand the transition out to the flowering. In the flowering pathway, just refer this image, because in this image they mention many protein names. Apex protein, apex as a major protein which helps for the flowering process. This transition of flowering involve the multiple factors. That factors are the gibberellin, leaf number, low temperature. If there is a the plant producing the the larger quantity of flowers and larger size of plant. This is the pathway which major role playing in the flowering. F1 protein and SOC protein, FLC protein. These are all proteins in the flowering. Here in this flowering, the floral meristem, floral leaf perimordium. These are the portions in the flower formation. Influence meristem also there. 4 factors involved: gibberellin, leaf number, temperature, and sucrose. These are all the 4 factors involved in pathway. Portion of the flower induction is the leaf perimordium, peripheral hemostatic gene, the floral hemostatic gene, the floral meristem. These are all the portions which helps to induction. Some maturation of the flowers after receiving the gene expression. That maturation of the flowers happen in the plant flower development, proliferation, differentiation, and that is all about the Arabidopsis thaliana, because Arabidopsis thaliana is a very defined, a very well studied plant. So many of the researchers are explained with the Arabidopsis. Okay, this image helps to understand the root and first one is the root and shoot development, and second one is the flower development. In this flower development, we having the different portions, the latitude cross section and the cross section one. Latitude sectionate one. In this we can see the stamen, capex, carpel, and petal, the sepals and vascular tissues. And the another one is the cross sectional image. This is, I'm going to end this now. Sorry for the this inconvenience, because tomorrow I'll try to fast in finish this portion within 1 hour. Thank you for your cooperation. I'm this section is open for the question and answer. We have lots and lots of question. Yeah, okay. These are the tomorrow topics: the molecular biology and recombinant. Now we are end up the 5th unit. The tomorrow we discussed the 2 portions of basic concept. Today we discussed the morphogenesis and organogenesis. Now this is going to be end up with the 5th unit. It's a developmental biology. Today I will send the questions, PPT and materials to your mail ID and in Google Classroom. Then tomorrow onwards we are going to discuss about, we have a 4-day more in the timetable. In this 4 days we are going to discuss about that molecular biology and recombinant that methods in biotechnology. That is the 13th unit. From the 13th unit we are going to discuss some topics. Tomorrow I'll try to finish and finish at the 1 hour, in the 1 hour. Thank you everyone. Now I am going to discuss the questions. I will answer the all questions in your Google Classroom and mail, because many questions are there here. Okay. Okay, try to answer here now. What is PSC? I'll send the definition to your Google Gmail ID or Google Classroom. Plasma cells are present in uh, the bloodstream. Dictyostelium is individual cell. That individual cell forming the group for cell development. Mold. It's one cell observing the signals from the bacterial and external source, and that cell forming the organs, and forming the network, forming the an external part for the mold. What are the protein gradients? Protein gradients, the gene presence presence in the particular embryo, but that is helpful for the organelle protein development. The protein is, if in one embryo that having the higher concentration of higher concentration of the skin tissues, that child may be developed with the higher weight. If there sometimes the gradient may have that gradient helpful for that parts production, the organ production. Satyabhama, I don't know the answer. I'll tell it in. I'll search the answer and tell it in your mail ID or Google Classroom. Why same colors are dark and same as light in diagram? Why? Okay, the color of diagram is explaining the expression of protein. If there is the if there is there they have the higher protein expression, that particular mold is been higher. The male and female has different abdominal segmentation. Yes. What is the pattern in the prochartrate like Amphius? Ishwari ma'am, I'll tell the such the answer and tell you, because this topic is new to me also. I want to learn something about the this. In regressive, the glial, the teeth and or shade of forms the tadpoles. I'll answer the questions. Better I'll answer the questions in the Google Classroom, because many of the questions are new to me. Sorry for the inconvenience. I'll send the answer to your mail ID. Now I'm going to end this section. I'll explain the answers in Google Classroom or mail, because this questions are very new to me. Okay, Arabidopsis thaliana, ABC model of flowers development in Arabidopsis thaliana. I'll send the notes for this topic, Satyabhama. Thank you everyone for your patience. I'll Send the answers to your Google Classroom and mail ID within this today evening.